So Punnett squares really show sexual reproduction. There's just ways of demonstrating sexual reproduction and showing the phenotypes and genotypes of the offspring, the percentage of phenotypes and genotypes. Dihybrid, di means two. Dihybrid crosses show crosses for two traits, and they're a little more complicated than the monohybrid cross. Review, if we're looking at monohybrid cross, that's a cross for one trait. Mono means one. So for example, let's say I'm just going to use a gene A. Okay, I'm just making up a gene, I'm going to call it letter A. Okay, we have one individual is big A, little a, heterozygote. Let's say one individual is little a, which is called a homozygote. And remember the recessive and dominant genes. So if it's a big letter, capital letter, that's a dominant gene. If it's lowercase, it's recessive. So two of them is homozygous recessive. So we're crossing a heterozygote with the homozygous recessive. Now, in sexual reproduction, we're shuffling genes. So sexual reproduction is like shuffling cards in a card deck. Crossing big A, little a, times little a, little a. Okay, then we're going to figure out what possible genes you can get from this. We can get big A, get little a. What possible genes can you get from this? You can only get little a, little a, little a, little a, little a, big A, little a, little a, little a. So this is a monohybrid cross. This shows the percent probability of the offspring having genotypes and phenotypes. So if we're looking at the genotype, that's the form of the genes. So that's what, like if it's heterozygote, if it's homozygous dominant, if it's homozygous recessive, that's genotype. So if I'm asking, if you cross these two, we're going to look at genotype percentages. I could say what percent of the offspring are going to be heterozygous. Well, hetero means different, remember? So big A, little a, that's different. Those are different. Big A, little a, they're different. Heterozygote. Same thing, this is a heterozygote. So 50% of the offspring, heterozygote. And 50% homozygote. We're going to look at dihybrid cross. So that's a cross with two traits. Di, two. These two. So here we have two different genes again. Okay, we have a gene and a gene. And we have two forms of a gene, big A, little a. So we have two alleles. The big A, the little a. Those are the alleles of gene A. Then we have a different gene, gene B, that has two alleles. Big B, little b. So we're going to say this is complete dominance, both of them. All right? We're going to cross these two. Now to do this, we have to use a Punnett square, but we're going to use a Punnett square with 16 boxes. When we set up a Punnett square for a dihybrid cross, it should look something like this. So it's 4 by 4, a total of 16 squares. Okay, we're going to look at the cross between big A, big B, little b, times big A, little b, little b. Now, doesn't matter which one's male, which one's female, since this is not sex linked. Okay, so I'm just going to take the big A, big B, little b. Let's just put that one up here. Doesn't matter if I put that one here or on this side. Over here, this one's going to be big A, little b, little b. Okay. So now I have to write down the possible combinations I can get from this individual over here. So we're looking at the gametes, in other words, the egg cell or the sperm cell. So remember foil, remember what like we used before, first, outer, inner, last. So the first A and the first B. That's big A, big B. I'm going to put that right here. Okay, outer. Remember foil, first, outer, inner, last. So outer A is big A, outer B, little b. Big A, little b. Inner. Inner A is little a, inner B is big B. And then last is the last A and the last B, which is little a, little b. Okay, so these are the gametes now. So these are the sex cells. For this, since it's not sex-linked, it doesn't matter if I put these on this side or this side. Now though, I'm gonna do it over here now. So first, outer, inner, last. You'll notice for this one, they're all gonna be the same because they only have big A and they only have little b. There's no other combinations here. So first is gonna be the same as the outer, the inner, and the last. Remember, we are looking at, so remember we need one of each of these genes, okay? So now we set up the Punnett square. That's the hard part. The easy part now is just showing the cross. So it's just down and across. So we're going down and across. Now you want to keep the A's together and the B's together. And remember again, capital letter will come first. So big A, big B, little b. Okay? Now you can do it by column or by row. I just go by row. It doesn't really matter. So down and across, big A, big A. So big A, little b, little b. Okay, now we have big A, big B, little b. Big A, little b, little b. Next one. I wanted to just write them all out, but you can see we have, these are the offspring then. So these are the possible combinations that you can have in the offspring. So these are like the possible zygotes. Okay, so if I want to know what percent of the offspring are going to be double heterozygote. So in other words, double heterozygote. They're going to have one big A, one little a, and one big B, one little b. So we're going to have to, so we can see, we have one here, right? Here, here, here. So 4 out of 16, which is 25%, are double heterozygote. Okay, so that's the genotype. With the phenotype, I didn't tell you what the traits would be, so I can't really, if I said, okay, so if I want to know what percent would be double homozygous dominant, it would have all big letters then. Okay, so we have, do we have any? We don't have any. Why don't we have any? Because this one only has 2 little b's to give. So the offspring are always going to have 1 little b. Okay, what about the probability of having double homozygous recessive? And this is when they would all be small letters. Okay, that's not possible either because this one only has big letters for A. There's no little a at all to give. Okay, so this is just an example of using a dihybrid cross, creating a Punnett square using a dihybrid cross. Creating a Punnett square showing a dihybrid cross. But remember, the Punnett squares just demonstrate really sexual reproduction. So we're crossing, let's say this is the mother, this is the father, or this is the father, this is the mother, it doesn't matter for this. Okay, and then these are the gametes. So these are the possible egg cells, or the possible sperm cells you can get. And then these then are the zygotes, these are fertilized eggs, so these will be the offspring. And the square itself demonstrates the probability or percent chance you're going to have certain genotypes or phenotypes in the offspring.